Okay guys, I want to tell you about the important parts of your photosynthesis lab tomorrow. One, you're going to be using these spinach leaves. As you can see, it's got some nice holes cut in it already. Who knew that a paper hole punch could do more than just punch holes in paper? Anyway, you're using these spinach leaves, and you're going to introduce them to a baking soda solution. That's what I have here in my beaker. Along with the baking soda solution, there is actually 1 drop of this Ajax liquid hand soap. The reason for that you have to have detergent, all leaves have a waxy cuticle, a waxy coating on them, and the detergent will help to basically break it down so you can get other things inside the leaf tissue itself. We're going to use these syringes here, and basically with these syringes you're going to be creating a vacuum. The tricky part is you guys get to figure out how to get the leaf discs into the actual syringe itself. I'll let you guys figure that out tomorrow. What I'm going to show you on the video is just I'm going to draw up some of the baking soda solution. Right now, all of my leaf discs from the spinach, right now they're actually all floating, as they should be. What I'm going to do is I'm going to create a vacuum right here. I see a bunch of bubbles going on. I'm holding the vacuum for about 10 seconds. Okay, I'm going to release and kind of shake these down. And I actually did this once already, so this is actually the second time I've done it, and it looks like I've accomplished what I needed to already. But you'll have to do this a couple times. Some pretty crazy science is actually going on inside this. Like I said, when you create the vacuum, you're forcing out all the air of the inside, the spongy mesophil, if you will, of the plant tissue itself. But as soon as I let my thumb go and I release the plunger, as soon as I do that, then I've now allowed my sodium bicarbonate, my baking soda solution, to fill that empty space that was once there. So now I've actually sunk these leaf discs, which is kind of cool because originally they were floating. Now the next tricky part of this, you're going to have 2 smaller cups here, and you're going to fill your baking soda solution into those cups. Again, another fun part you guys get to deal with tomorrow is now getting these leaf discs out of the syringe. Easiest way I found is actually just kind of dumping them into somebody's hand, and you guys can then distribute them equally into each cup. One of these cups you're going to cover to basically kind of show that sunlight's kind of needed for photosynthesis. And then we've got these lamps. So I'm not going to turn the actual lamp on because it's really bright in the video. But you'll expose one of these to the artificial sunlight. And so basically you're showing the light-dependent versus light-independent reactions for photosynthesis. One really important thing the other thing I want to make sure you guys understand is with the baking soda solution, try to zoom in a little better. Baking soda itself, with the actual chemical formula, is NaHCO3. What's really important about that is that bicarbonate ion, the HCO3 ion. That's your carbon dioxide source. So as soon as you basically infiltrated the space of your spinach leaf with that baking soda solution, you've introduced carbon dioxide to the inside of it. Hopefully, when exposed to light, that carbon dioxide, which is a reactant for photosynthesis, will then be a chemical reaction will take place, and hopefully then oxygen gas will be produced. And with the oxygen gas being a gas and nice and light, the goal would be to see your leaf discs floating once again. That's what we're after for tomorrow, guys.